Hello and welcome everyone. I'm an Injanja, a data journalist and communication specialist at the Center for Epidemiological Modelling and Analysis, CEMA, at the University of Nairobi. And I'm the session's moderator. So today we are starting with a biological riddle. In the Middle East, this virus is a headline-grabbing killer. In Africa, the animals carrying it are everywhere, millions of them. Yet the words remain quiet. Where are the cases? To answer that, we are diving into our session today, the African MERS-CoV paradox, high camel exposure, low human cases, unraveling the missing links. To lead us through the science of this silent threat, we are joined by the newest doctor in town, Dr. Ogoti Maina. I say new because Dr. Ogoti successfully defended his PhD yesterday, earning him the title. Congratulations once again, Dr. Ogoti. So, Dr. Ogoti is a virologist specializing in infectious diseases, combining wet lab pathogen characterization with computational methods to understand disease transmission dynamics. As a postdoctoral researcher at the Center for Epidemiological Modelling and Analysis, it contributes to the pool of research in emerging zoonotic diseases in Africa. So today he will unpack what MERS-CoV is, break down the infection patterns between species, and more importantly, explain why African continent isn't seeing the outbreaks many expected. Please use the chat to introduce yourself, to post your questions, and to learn more about CEMA. Dr. Ogoti, welcome. The stage is yours.
Thanks, Annie. Am I clear enough for the audience?
Very, yeah, you're clear, you're audible.
All right. Thanks everyone for joining this CEMA Africa seminar series. And today I want us to just stray a bit from to look at a different thing, different biology of a different silent threats that over the last 3 or 4 years I've undertaken as part of my PhD in immersed in this one for a while now. So just to give you a recap, thanks Annie for the excellent introduction. I am Ogoti and I'm a postdoctoral fellow in CEMA. So I always love this acronym MASC, which means Middle East Respiratory Syndrome Coronavirus. Coronavirus is not a new terminology among us. The recently concluded SARS-CoV-2 pandemic was quite had high mortality and morbidity. And interestingly, to tell the audience today, this virus actually was discovered earlier before the recently concluded pandemic. I'll just take you through the virus, and then I'll give you the overview or the links, in my opinion, or as per the research that we have done, of what we actually think of this virus and what are the future implications of this virus in terms of human infection. So, as I said, it emerged somewhere in 2012, saying emergence, that was when it was first detected by a very brilliant doctor in Saudi Arabia. So this patient came in with a known etiology, but he was presenting with actually acute febrile illness, or we call the SARI case for the clinician to the room. They tested for all known pathogens to that time. None was turning positive, and then unfortunately, this patient zero died. But when they actually did virus isolation, they actually realized it's a coronavirus, but has not been characterized before. And then that's how the name emerged. To date, we see up to around 2,600 cases with around 900 fatalities, with actually a very high case fatality rate. This is actually even higher than what we have seen from the recently concluded pandemic. And of course, I will explain why that's the case. So far, it's very endemic in the Middle East, but cases have been imported to other countries. And specifically, if you look at the map on the bottom left, that's shaded in red. Those are some of the countries where cases have actually been reported. Looking at the epidemiological timeline, from 2012, since the first case was detected, I'll just drop my pointer to make it easier. You can see a very big spike of cases around 2014, and this was actually an imported case to South Korea. So an imported case in a hospital in South Korea led into one of the greatest, highest number of MERS-CoV recorded outside the Middle East from patient to patient. As you can see, this epic curve, somewhere in 2012, we actually have low cases. This actually is not, in my opinion, as an expert on MERS, it's just that what happened in 2012, most resources were allocated to the recently concluded pandemic. But so far, the cases are actually now picking up, and there are some reports. And what is the transmission range, if you ask me? We see a very quite interesting picture that in the Middle East, we see camel to human transmission, and I'll explain why the camel is actually here. In Africa, predominantly, we see what we call a camel to camel transmission. And as I highlighted before, some countries like UK and South Korea, that's an Asian continent, we had some imported cases where there was a hospital to hospital nosocomial transmission. So that begs the question, where did this virus come from? And if you look at this phylogenetic tree, which is actually a very nice tool as virologists and biologists to use to actually differentiate different microorganisms. So we want to understand, are things related? And this is actually the reason why. And just to interpret this thing for the audience, when things are actually originating from one branch, this is what we call a branch, they have some sort of similarity. So it's like a puzzle. The closer you are to something, the more related you are. And in this case, the initial mask of samples, when they were actually sequenced and plotted to a very close coronavirus isolated from a bat called Neuromica, we could see actually some very close similarity. And that makes the question. So we have seen the bat is actually really close to the virus. It was first detected. What is this missing link that actually sustains this virus in the environment, or actually in the whole cycle of the virus? And that's why I have this very nice animal. It's called the camel, but the scientific or the species name of interest for us, there are different species of camel, but specifically the dromedary camel, which is actually Middle East. And this is supported by many reports. And if you can look at this timeline, these are some of the studies that have been done. Sorry, Annie? Okay. So this is just a timeline of studies that have been done in both Africa and the Arabian Peninsula, in black and red in Africa. So you can see these studies have actually characterized there's actually a widespread circulation of MERS in dromedaries for a long period. In fact, the earliest reports are from Sudan and Somalia by Muller et al., which showed neutralizing antibodies camels, which kind of gives you the idea that even though the first case was detected in humans in 2012, we have actually a widespread of this virus in the reservoir host, that's the camel, for very many years. And of course, this virus, if you look at it from a virological point of view, it's divided into different clades. The one in red is the clade which is dominant in the Arabian Peninsula, which is clade B. And then you have clade C, which is found in African continent. And this study by El Karafi et al. In 2019 just deposits a phylodynamic relationship of all the available genomes that were sequenced from then to 2019, and trying to see is there some form of interconnectedness? And actually, you could see that there's some form of flow of the virus from the West African continent, and you can see there's actually a big concentration of this signal in the northern horn of Africa, and actually that's how it exits to the Middle East. Middle East, and then that kind of begs the question: if this virus is circulating very successfully in camels and especially in Africa, why are the zoonotic or why are human outbreaks actually restricted in the Arabian Peninsula? Just before we dive into whys, I want us to go just what do we know about MERS-CoV in Africa? To date, WHO reported cases are only 6. So only 6 human cases, and all those cases had some history of travel. Interestingly, when I say case, it does not mean disease. The cases in Africa actually had no, the primary cases had no indication of disease. If you compare that to the Arabian Peninsula, we see over 2,000 cases and with a case fatality of 36%. The prevalence of the virus in camels in the two continents, it's actually very similar. That is the paradox, where that's a paradox fact, that this virus, if you look at the prevalence in the host, it's actually very similar in the two continents. Another interesting fact which will build up the discussion is this actually a unidirectional trade of camels from the African continent to Middle East through the port of Riyadh. And just going back to the exposure, if you look at in terms of seropositivity, or if you look in terms of other immunological markers, we can actually see that one study in Nigeria did the two, and actually they could see differences. Whereas these camel abattoirs, who are actually a high-risk group to exposure to the virus, had almost zero seropositivity. They could actually detect up to 30% T-cell positivity, which is a different marker of showing exposure to MERS. So far to date in Africa, we have only 3 RNA positive cases by our colleagues in Washington State University in Garrett et al. They describe 3 cases active. That's what you call RNA positive, but they didn't have any history of exposure. And then finally, as I described, there are 2 variants and distinctly different. So the quick questions come: what actually is the infection patterns? And in this case, since our study, or in this study, we decided to do it in Kenya, which is a part of the northern horn of Kenya, what are the infection patterns of MERS around this part of the continent? And also the most pertinent question, why do we see limited human cases? For that, we and my colleagues, we undertook a systematic review, which had some quite interesting highlights. And this is just an example. We were able to actually get 53 relevant articles based on our search terms and our research question, because we wanted to understand what is the epi of this virus, and specifically epi and the genomic features in the whole of Africa. We also did a meta-analysis, and in this case, we found 31 relevant articles. And this is just the distribution of articles. And I decided to distribute them based on the host, and sometimes these articles were actually overlapping. And if you can see the most interesting part, most studies actually originate from the northern horn of Africa. In fact, if you take Egypt, Sudan, Kenya, and Somali, they contributed at least 2/3 of the whole literature for understanding this virus. And it falls very nicely because we all understand that most of the camels actually reside in the northern horn of Africa. In some of the results, just to quickly show you, is that we looked at the different frequencies. That is, one, when I say MERS-CoV RNA, this is just describing a precursor of what we call an active case, and anti-MERS-CoV IgG is just the exposure. And further, we actually categorize these 2 different hosts in terms of age. So specifically for the dromedary. So we had 2 groups, the adults and juveniles. And in this case, juveniles are described as dromedaries below the age of 2 years. And you could see some different or just some random patterns, but what actually most is, if you look at the adults, they're quite exposed with very low RNA positivity. And actually, although the juveniles have quite high exposure of the virus, seropositive for the virus, they actually also add higher RNA positivity, and that will fall very nicely in the next discussion. Just to complement the results, we did a full meta-analysis to see what are actually the absolute values. And if you look in terms of seropositivity in these two hosts, if we start with dromedaries, in fact, you can see dromedaries are quite highly exposed, and this falls very nicely from the primary studies that I've been reporting. The full prevalence of this is around 70%. So dromedaries are quite highly exposed, and this is very different in terms of active infection from one most study actually, which is around 6%. Of course, humans, you don't see active cases, and that's why you have zero. But interestingly, for this paper, is that you saw up to 2% of humans actually exposed to the virus. Further, if you look at the subcategories which you decided to do, either age or region, you could see different features. But the key message that was actually one of the highlights of the paper is, if you look at adult camels and juvenile camels, they actually have a very vice versa effect. Adults are highly exposed at 80%, which is very different from juveniles at 30%. But this is very vice versa. When you look at active infection, juveniles are at 15% to date from all the studies that have been done, while adults are at 4%. And this quite falls nicely because we know juveniles post 6 months of life, they wean off those necessary antibodies for protection, and they become susceptible. Just to finish off this review, which I will link, which we'll have the link in this, after this seminar, is if you look at the genomic features, and just to take you through, because so that you don't get lost on this plot, this is just a plot of how the genome, a representative genome of MERS-CoV will look like. This is just a standard. So we have this what we call structural protein. And we could see some unique polymorphisms that the African variants have collected so far from the date when they were actually detected. So we see some interesting polymorphism, and some of these polymorphism at this point are quite inferential. So we can imagine what it's trying to do, but this could be an hypothesis. One of the reasons why we actually see an attenuated phenotype of the virus in African continent. Just to take on to the discussion to the next level is, after doing our review to understand what is the burden of the virus continent, we actually set out a very interesting field study, which actually was my work over the last year. It's called ZUTRAM, which is an acronym for zoonotic transmission of MERS in Kenya. This is just a map of Kenya. This is where we sat, a county called Isiolo. And specifically, we sat in a very specific place. It's a camel slaughterhouse, which we have very nice pictorials here. One of the years, I don't remember, it's around 2023, if I'm not wrong, where we actually set out to do a very specific objective. So one is, instead of following specific herd of camels, which has logistic challenges, and also you are not so sure if it's representative of the region, we sat in a camel abattoir and allowed the camel to come to us and sample them. So over a period of 12 months, we sampled 10 to 12 camels daily, which gave us around 2,700 camels at the end. The interesting part about this camel is that camels come all over northern Kenya from the neighboring counties, and they are either transported to the abattoir through by foot, or sometimes they're brought through lorries. And over a period of 12 months, we're able to actually pull this off. And this is just an example or a picture of the camels actually resting before the slaughtering process. The other features of this camel was actually interesting is that it's an open slaughterhouse. It's not a closed system slaughterhouse. And this is just an example. There was some open sewages and some of the birds taking the remnants, and there are actually some cattle grazing around that. So This is just myself somewhere around late 2022 doing a PCR just after collecting the samples. So we collected serum and nasal samples from the camels. And this is just an exciting result of my first detection. So this is an example of a screen of how a detection amplification curve in PCR happens. And I had my first 2-3 samples, and it got me PI of this project, which also from Professor Tumbi Mwangi, giving a lecture somewhere around the same around the same. So what are some of the results that you saw? If you look at the metadata, the camels originated from all over the northern part of Kenya. And in my testing, I was able to detect 36 out of 2,700 RNA positive camels. And this was just the distribution of, based on the origin of camels, the percentage. But most importantly, which forms part of our other paper, which describes the biphasic incidence of mass of dromedaries in northern Kenya, is that we see two peaks across the 12 months. So the first two peaks occur in September 2022 and October 2022. Then the cases die off. And then we see them again in January 2023, February, and March. And then they actually die off till the end of the 12-month study. And this kind of gives us a very nice idea that this virus does not endemically circulate throughout the year, and it follows some sort of pattern. And when actually we overlaid this data with environmental factors, specifically rainfall, because we know the dromedaries actually move around for forage and water, especially during the dry season, we could see actually a very interesting finding that we could see a negative correlation of rainfall and cases. Just to expound that on more, is that whenever we saw less rainfall, actually that's when we were detecting the cases, and vice versa. Whenever there was more rain, we could not actually detect any case. And this is actually very intuitive because we understand that during the dry season, the camels congregate in waterholes, they come together, and this actually becomes a transmission event. And during the wet season, as now in Kenya, is all these herds actually go back to their origins, and then you have this risk of transmission. What about those people working with these camels? We recruited all of them. There are actually 48 in number in the whole app, and decided to look actually at these people exposed. We tested them by PCR, none were positive, but if you look at their seropositivity profile, we found that 7 out of 48 workers actually had elevated anti-IgG responses. Just to confirm our findings, we actually had 1 out of the 7 responses being confirmed by neutralization test, specifically a neutralization test, which we actually, how's it called, in-house developed during my studies. It's called a VSVPP. It's a pseudotype. So imagine a virus which carries another part of a virus, and that's what you use to do a neutralization test. And I will not go to the technical details of that. This is the study. I will link it in the description. And as part of this work, we were able to actually do some trainings for the people. Then it was this at Unity, just trying trainings of people around Kenya on agnostics. So another thing that we actually realized over time is that camel slaughterhouse is very nice. Yes, you actually sit and wait for them to come from all over from our catchment region. But one of the limitation of an abattoir, especially, is the limited infrastructure, especially the silo one, it's an open air. So, what if use of rapid test, and this was actually a very good tool during the pandemic. Can we use rapid tests actually to detect outbreaks before the confirmatory tests like the gold standard PCR are done? And for this, we actually validated a very specific commercial test for MERS-CoV in this same population of camels. And just to go through quickly what the idea around it is, this test the principle around it is you have a nucleocapsid, which is detected in the using monoclonal antibodies in the strip, and then you have and it's similar from the same principle of what we saw in SARS-CoV-2 test. So we wanted to understand, is there differences in terms of virus? Because we know there are different variants in Middle East and Africa. We see a difference, and then we did the validation, and also we actually piloted it in a prospective study. So the first thing that we did is to check, because I told you it's based on the nucleocapsid protein of the virus, is to check, do we see polymorphisms between those variant groups? And of course, yes, there are actually some polymorphism if you compare the reference Middle East versus African one. But most interestingly, when we did the test, is that when you see the clade A, which is a representative virus from the Middle East, and clade C, which is a representative virus from Africa, we could see that we see no significant difference in terms of how they were tested. And remember, these samples in the background, they were confirmed by PCR. So that is our gold standard. We're actually validating this test towards. Of course, we see some bit of difference in RNA copies per mil, but this is because of we hypothesize the defective particles that are generated from the virus. But most importantly, when we tested this test from all the samples we collected from the first part of the study, around 36 of them positive, we could see a clear difference. So the limit of detection of the test was around 10 to the power 6 RNA copies per mil, which was actually very nice, because if you look at the positive, they have different colors. So we have these solid reds and the ones which are hollow. Some of these samples were actually, we attempted virus isolation attempts. And it was actually very nice to know that all the virus isolation attempt samples were actually detected positive by this kit. And this will form a very nice message at the end. And looking at just the basic parameters, this kit has very low sensitivity, around 25%, but it was very specific. So it could actually distinguish true negatives. But of course, the sensitivity was quite low, which is expected because PCR is actually a very sensitive tool, a very sensitive test. Just before I go on to the final or the major part of this discussion, or just unraveling the links, is the usefulness of this kit, despite the poor or the low sensitivity, is it can actually distinguish an infectious state. And this is very important in outbreak response cases. This kit, of course, cannot give you all the possible positive based on a test like PCR, but it's very useful in distinguishing outbreak, because it can actually distinguish all infectious samples, which you know they have almost over 10 to the power 6 RNA copies. So the other part of the discussion is we wanted to understand, why do we see limited human cases in the continent, looking at the scenario in our country in Kenya. In this part, we looked at what we call T-cell responses in these workers, in abattoir workers. A very nice gentleman who I'm sure is in this meeting, called Victor Ido, was very instrumental in this work. And what we actually set out to do is look at spike peptide pulse. When you look at the virus like MERS-CoV, they have this glycoprotein projection, which are very nice, especially during virus entry, and also they are targeted during vaccination or by developing vaccines towards them. So we stimulated this with blood from these abattoir workers, and we actually did 3 different screens. In addition to using MERS-CoV peptide, we also included other peptides like SARS-CoV-2, because of course it was widespread by the time we were doing the study, and also we tested all these samples for MERS-CoV IgG. This is just the different screens. This data is still unpublished, but you could see that we could see a detection signal based on interferon gamma responses. That is a precursor of a T-cell response to this virus. But most importantly, what I want to get out of this study is that when we look at the antibody responses, which is represented by this curve, which is called the odd duration, and when you look at the T-cell responses, which is mentioned by interferon gamma, we could see no correlation. Like an example, this sample, which was highly positive based on interferon gamma responses, had quite low antibody response. And this is quite nice and has been described before by other MERS-CoV researchers. Is we hypothesize that the virus circulating in the African continent actually causes what we call subclinical infection. And in the cases of subclinical infections, you always find that the antibody responses or the yield of the antibodies is actually not sufficient. Also, another good explanation of this is antibodies actually wane over time. And we have these T cells, which are memory cells, and they quite are sustained for a long period of time once you are exposed. And of course, this is just another plot showing this. When you do a correlation analysis, there's no correlation between interferon gamma responses and antibody response. Just to finish off, and I can come there back during the question and answer. Question is the big chunk of it is because this virus is different, at least from the genomic point of view. But do we see a very different phenotype? And for this, we actually got 12 full genome from the 36 positive samples. This is just a phylogenetic tree as the one that I described in the previous part. And as you can see that our samples actually cluster somewhere here in a subgroup called C2.2. Our nearest neighbor is sample. Of course, this is Djibouti, but the updated trees, the closest sample is another sample that was isolated in Ethiopia. But most importantly, just to understand this, if you look at previous samples isolated from Kenya, they're quite far away from us. And this is interesting. And this is just a period of 2019 to now, a period of 5 years. You can see that these variants, especially from this part of to able to isolate 6 viruses, and this work was done in Adite together with a very, very nice gentleman called Dr. Jody. Just before I go, these variants, if you look at our Kenyan, we could see different polymorphisms. And just an example is we saw 8 new spike polymorphism that have not been described before, and also the structural proteins have some issues, which builds up to the story that this variants in Africa might have some form of attenuation, and in part actually may contribute to why we see actually very low cases. And I indicated that this virus is isolated. And if you look, this is just a plot for the non-virologist in the meeting is, in the lab, we can actually isolate a virus and try to mimic its growth kinetics by growing it in relevant cell culture models. Like what you could see is actually lung cells, specifically lung cancer cells, which have been modified in the lab for us to help us study the virus. On the X or the X-axis, this is just the time. So if you grow a virus now, monitor it over a period of maybe 24, 42, in this case, we went up to 72 hours. And what you understand is, in the orange are representative Kenyan virus shows, but if you look this against the references virus that are in the Middle East, Cladian B, you could see that if you look at my y-axis, which is genome copies per mil, which is a representative, how much amount are we seeing of the virus over time? You can see the amount of viruses that we see from the Kenyan virus are quite lower compared to the Middle East variants over time. And this was actually, if you can see this graph, presented nicely, alveoli cells, human alveoli cells, which we in vitro use in the lab. And you can see that the growth kinetics of this virus is actually quite limited compared to the reference viruses that are actually causing human infection in the Middle East. We compared this also with another relevant model, which is called human bronchial culture. Depiction of virus growth, and this one circle represents a well where cells been grown, we use a certain dye just to visualize the effect, because this virus actually live by causing either cytopathic effect or by lysis. And you can see this compared to Saudi Arabia actually quite compared to the Kenyan viruses, which are quite low. And this quite gives us an indication that the viruses that circulate in the African continent might be actually attenuated, and in part actually might explain why you see these non-human cases vis-a-vis the Middle East continent. And this is just the same plot, this hatchback, same plot, just showing that Kenyan viruses uh, replicate at lower titers compared to the Arabian Peninsula. Just in summary, to go through, because I've gone quite from the field to the lab, nation, just to give you 3, 4 key summary points. One is the incidence of MERS, especially in dromedaries in the African continent, might be seasonal. And this is actually very nice. And if you look at various studies by our colleagues, is that you consider there were specific detection points at a period of a season. And the second part is the viruses, especially the ones detected in Kenya in the last 2 years, have subclated themselves in a clade called C2, and it seems to be the dominant. The third point is, if you look at the transmission dynamics, MERS-CoV transmission, there's actually likelihood of this virus transmitting to humans, especially to the ones we call them camel exposed or risk individual who have exposure towards camels. And in this case, we actually detected both parts, the seropositive antibodies and also the reactive part of just another marker of explaining exposure towards human. And then finally, one of the links that you actually get out of this study is the virus or the clade C, which is currently circulating, seems attenuated compared to the references virus in Middle East, that is Saudi Arabia and other. And most importantly, these strains in the continent must be avoided. This statement we recorded it somewhere last year when some of these summary key points. But to tell you to date, there's one study that has been described in Egypt that has shown a clade C virus has actually recombined with a B, and it's actually not classified as a C anymore. And this is why actually we really ought to have this surveillance by surveying risk individuals and also doing sentinel surveillance in dromedary to understand the circulating variants. As we know that the B actually causes human infection, that's what we want to avoid this in our region. I will acknowledge very different people here from Jody and Kani for setting up. Also, my mentor, Professor Marcel Muller, was actually instrumental and is a PI in this project together with Tumbi. And also the people in CEMA, including Patrick, was very good in doing the studies or the sampling of the camels over the period of 12 months and also the counting. And also we can't forget to mention the funder for this work, which is the DFT project. Yes, I will open the floor for this for questions or any other comments that you'd like me to clarify. Thank you very much.
Thank you, Brian, for that informative session. And we open the floor to questions. Please raise your hand, unmute yourself, or post in the chat to start us off. Okay, and as we, sorry, somebody wants to post a question? Okay, as we wait for guys to post in the chat, Brian, what does this mean in like looking into the future? What does it mean for the next jump from animal to human?
Thanks, Annie, for the question. There are different ways to look at it. One is At the moment, and if you download the latest WHO risk assessment report, is that the risk at the moment is low because we don't see the virulent strains of Middle East introduced in our hearts. But more alarmingly, as I spoke during my summary slides, is that we see one study where Camel in Egypt was isolated with a virus which has some recombination signatures with the clade B. So there's actually a risk of this virus C recombining with B and having that introduction and hypothetically causing increased human infection. Of course, to date, we have had the privilege having, if you remember in my initial slide, the camels are actually directionally traded to the Middle East and not vice versa. And that's why we hardly see the viruses or the camels in the Middle East coming back to the continent. But there are different ways to look at it. One is that these extracurricular sports that are happening, not at the moment, but we know that are happening, including camel horse racing, camel beautification, and these events actually might at a point end up in somewhere in our continent. And in the case these viruses are actually being carried, they find a way to spill over to the local herds. That's actually a risk. But at the moment, we see the virus attenuated, which is actually very good in terms of public health. But most importantly, ought to develop preventative all surveillance strategies, especially to monitor the variants. And this was actually a very big topic in our recently concluded pandemic, variants of interest. But in this case, we ought to actually have a sentinel surveillance, at least surveillance of the host to look at the different variants of interest. Secondly, there is actually a development that there's a mass vaccine available. It has gone through the phases of trial. It's being tested in humans now. It's a vector vaccine called MVA. And the good news is from the preliminary results is that it cross neutralizes both variants. So even if you get the vaccine, it actually is protective towards the clade C or the clade B variant, which is actually a nice positive encouragement. But the idea is keeping up to just ensure that the circulating variants or the variants of interest are noted early enough so that intervention or control strategies are actually done.
Thank you, Brian. We have a question from Joki Kimani. She's asking if camel MERS-CoV prevalence increases during specific seasonal conditions, how should public health preparedness and One Health coordination be strengthened during these high-risk periods, in particular as climate variability intensifies?
Good question, Joki. And actually, I totally agree. We see the variants amplifying at very specific point of the period, but there is one big factor that is happening at the moment called climate change. So we are seeing irregularities in season, and we hypothesize that will have a ripple effect also in how the virus occurs and at what different points it occurs. My recommendation will be in terms of setting up a surveillance effect just to monitor these peaks is incorporating rapid diagnostic tests. So in our study, we actually do a rapid diagnostic validation, and we see that is actually good enough to detect infectiousness or acute outbreak camels. So I will actually recommend this use of the rapid test to actually detect acute outbreaks in the field. It can be done on site. It doesn't require technical or a lot of technical expertise knowledge. So it can be done literally by anybody with literacy knowledge. So that's actually the positive part of it. But generally, I think pinpointing specific months, that will be difficult. And also to add on the point is climate variability has come up with a very different curveball because most of these herders are actually diversifying by keeping camels. Traditional cattle or goat keeping people are now moving towards camel. One, because the value of the camel is high, it fetches quite a lot of money in the market. And an expert of camel value chain, I'm sure is in the meeting, Pauline Gitonga, can shed a lot more light on it. but there is interest in camel herding and increasing these activities, and this will increase more chances of human-camel contact in the coming months or years, and this will be better. Thanks, Njoki, for that.
Okay, we still have some time left. If anyone has a question, now is your time. Njoki appreciates the response. Then I guess that means the session was very thorough, and you are thorough, Brian. If there are no additional questions, thank you all for joining us today. And Brian for taking your time to really share about this topic, African MERS-CoV, and for unraveling that paradox, such a critical piece of the global health puzzle. And we thank you, and thank everybody else for those who have asked questions. Thank you so much, and everyone else for joining us. We will post this recording on our YouTube channel, and feel free to check within the week. Thank you so much, and have a nice week. Thank you.